Right then, ladies and gentlemen, now it's time to start the exciting part of the meeting, where those people who are actually doing the work get the chance to present something to us. And your job in the audience is then to give them a hard time in terms of questions. A number of points about this. The posters will actually be displayed for the next 2 or 3 days anyway, so there's plenty of time to look at the posters, see the details, and ask questions of the presenters when you've read the details. Another thing to say is that the poster sessions, and especially the poster prizes, the very generous poster prizes, which will be awarded if any of the presenters actually deserve them, these have all been donated by MacuHealth, and we must thank MacuHealth and Frederick Juhay for this generosity. Thank you very much. Now, the way that this session is going to work is that each presenter is allowed 3 minutes. That is 3.00 minutes. And after that, we're allowed 2 questions for each speaker. Any speaker who is still trying to talk after the 3-minute time will have a card waved at them. If that doesn't work, Neve will shout at them. That will almost certainly work, but if that fails, this stage is fitted with a trapdoor, and the speaker will disappear. What happens after that, I don't know. But I'm not having sausages for breakfast tomorrow, just in case. So, let's get started. I'm not going to say the titles of any of these presentations because I assume that most of them will appear on the first slide anyway. But I will introduce the speaker. And our first speaker is, if I can find the right piece of paper, our first speaker is Nidhi Batiwada from India. We have a mixture of topics, a great mixture of topics, a lot of interest for everyone, as you will find out. And John is kindly offered to help me, and he is operating our very precise and accurate timing machine. So when you're ready, click the machine, click the speaker, and let's get started. Thank you, Nina.
Thank you. Good afternoon, everyone. I am presenting my work, that is Dietary and Lifestyle Risk Factors Associated with Age-Related Macular Degeneration. And this is, out of interest, we have done in India to compare the results from the Western as well as European and UK or US countries compared to what is happening in India. So these are my 2 objectives. I'll be looking into the risk factors as well as the dietary intake of lutein and zeaxanthin. This is an ongoing research. We are still, it's an ongoing work. We have selected some 3 to 4 hospitals. Out of that, till now, 4,378 subjects were enrolled, and around 3,500 participants have already participated. And these people have undergone ophthalmic examination, detailed ophthalmic examination, as well as for the grading of AMD. This was further divided into 2 steps. One was masked interview for risk factors by questionnaire, and secondly, the dietary intake of lutein and zeaxanthin as well as beta carotene was done by food frequency questionnaire. These are the results part. The AMD was found in 2.2% of the population, that is 77 subjects. Early AMD was 1.8% of the total population, and late was 0.4. These we can see the fundus photograph of each. There are fundus photographs for each individual which was used for grading. This graph will show as the age increases, AMD cases also increases. This is the association between AMD and dietary intake of carotenoids. We have done a chi-square test, and here we can see that mg per day, the lutein as well as beta carotene intake, and it has shown a very, very statistical significant result. And we can see that as the lutein intake decreases, AMD is found to be more among the patients, and same was in the case of beta carotene also. This is the bivariate logistic regression analysis. In this, we have seen the association with risk factors, various type of risk factors that can be lifestyle or dietary or medical history. We have found out age, diabetes, as well as smoking and alcohol whether it is moderate or heavy smoker has a odds ratio more than 1, and they were very much significant correlated. In the case of female, in this population, the odds ratio was more than 1, but the statistical correlation was not significant. The same was followed for hypertension also. When we have looked into the dietary intake of lutein, zeaxanthin, beta carotene in the logistic regression analysis, we have found that it was less than 1. So, we can say that the increased dietary intake of lutein and zeaxanthin has reduced the risk of AMD. And this is the conclusion part, and this is my institute. It's a Palace Converted Research Institute, and this is like smoking cessation and alcohol may reduce the risk of AMD, and risk of AMD is higher among the increasing age groups as well as the diabetes. Increasing the intake of lutein, zeaxanthin and beta carotene can reduce the risk of AMD, and these are the acknowledgement I want to give for my director, my mentor, my lab mates, as well as the hospital who has collaborated with us, as well as the organizers of this committee that has allowed me for presenting my work here. It was within 1 week. Thank you. It was on time, sir? Oh.
Yeah. Thank you. Don't run away yet. Don't run away yet. We have time for 2 questions for each speaker. Please, questioners, make the questions simple and no multiple questions, please, for time. So here we have 2 questions lined up.
Actually, sir, this is still we are coming up with data and collecting data, but whatever population we have collected till now, it's around 3,500. We have found only 77 subjects were cleared with AMD. That was 2.2%.
Did they have geographic or did they have?
We have made clusters, sir, in the city, and we have taken random clusters from there.
There's 2 types of AMD. Which type?
Early and late, both we have done.
Okay.
Early was in 63 patients, that is 1.8%, and late was in some 14, that is 0.4%.
In the late group, were they?
Okay, excuse me. Can I ask you, if you want to extend any questioning and concentration, please do so at the posters, because we are very pushed for time, but we can take.
That was an amazing job. In epidemiology, we always worry that how we select people to come into our study might influence our results. So have you thought of, how did you select people, and how do you, can you argue whether that would or would not influence your results, for example, the protective association with lutein?
Actually, ma'am, our study was in Mysore district. It's a place in India, southern part of India, and then we are divided into rural, that is rural part of Mysore city, the city part as the rural part, rural and urban. Then we have divided into different clusters, and then we have randomly selected basis on the health ministry data will be there. According to that, we have taken the cluster randomly.
And so, is it your opinion then that you've selected a representative sample and that you should not have bias?
For this, we have we have taken a help of a statistician who was working in that government of the health sciences. And he has helped us to take the representative sample. And still it is ongoing. We need to have more number of people to come out with the better understanding of the outcome.
Okay.
Yeah, but
thank you.
Thank you.
Thank you. Right, that was a good start. So now we move on to a presentation by Janet Frick from from the Hammond Lab in Georgia, United States.
Oh, okay, great. Awesome. Awesome, great, thank you. Thanks, everyone. My name is Janet Frick. I am a developmental scientist at the University of Georgia, and this project is a collaborative effort between my laboratory, which studies infant visual attention and cognition, and the laboratory of Randy Hammond, who you all know as a longtime carotenoid expert. So I am here to talk about the baby side of things, and if you all have any questions afterward, I'm going to have Randy answer them. I also want to acknowledge the work of my graduate student, Sarah Saint. Sarah Wade Acabei. This is her, is going to be the foundation of her thesis work, and also Kevin O'Brien was another collaborator. What we are trying to do in this project is to examine the early part of the developmental questions about the impact of lutein across the lifespan. As you all know, the effects of macular carotenoids on the adult brain and visual system are very well known. We know less about the impact of carotenoids early in the lifespan. However, there are good reasons to think that the impact of carotenoid intake may be as important or potentially even more important early in the lifespan compared to what we know about later. One of the behavioral measures that has been used as a way of assessing macular pigment is temporal processing speed. And what we have developed, we think, is a valid way to measure that behaviorally in infants. We are measuring critical flicker fusion threshold behaviorally in the infants in our lab. The way that we are doing that, and we'll be glad to elaborate more at our poster, we have a small sample so far. We'll be collecting a lot more data. We have built, and by we, I mean Randy and his minions in his lab, have built a custom device based on LED clusters. We have different shape cutouts to make the shapes interesting to the infants. And then the baby, we're doing a basic preferential looking task with the babies, where one side of the stimulus, one side of what they're seeing is flickering, the other side is solid. And a blind observer will watch the infant and judge which way the infant is looking, and will make a determination, are they able to see that particular threshold or not? We take a 75% correct criterion as saying that they were successful at that level. Using this technique, and like I said, our data is coming in. We plan to collect a lot more data as we go forward. But so far, we have a correlation with age of about 0.40. There is, as you can see in the scatter plot, and we'll be glad to talk about this more, there's a great deal of variability during early infancy. We want to try to study what are some of the predictors of that variability, and we do plan to collect much more data with older kids. But this procedure is well tolerated. We can do it in less than 10 minutes, and we can keep it as interesting as possible. Is it possible that this is linked to macular pigment as it is in adults? We plan to find out. So I am done. We think this is a great measure for babies. And our real question, this final point, I will let you read as you ask me questions. We think it may be related to feeding status. We plan to study formula-fed versus breastfed infants. Thank you.
Now, any questions, please?
Randy, you may have lucked out.
Anything to it?
There's,
Go ahead.
Yeah.
How do you measure cognitive measures in the baby?
that is, and I could give a whole long talk on that. That is really my background. But most of the work on early cognition in babies is based on visual attention. You can measure memory. You can measure speed of processing. Essentially showing babies pictures repeatedly and then adding in new pictures, measuring their amount of fixation to things that they haven't seen before versus ones they have. Another measure of that's shown very strong prediction with later cognitive outcome is you have a baby fixate a central target, a peripheral target appears, and you measure how long it takes them to disengage from that central target to a peripheral target. It's a behavioral measure, but it's been shown through imaging studies to be related to different brain systems that come online early in the early months of life, and then longitudinal studies following babies up into early childhood, middle childhood show a moderate correlation with later IQ and with other cognitive outcomes.
Have you got any results yet?
I'm sorry, sir?
Have you any results?
This study, we have not done longitudinal work like that, but that is part of our master plan. But yeah.
Thank you. What's the very youngest age that you could predictably get a result for?
Could do with this, I would say realistically 2 to 3 months. The it could potentially be done with newborns, but as anyone knows who's tried to study an attentional-based response in newborns, their window of alertness is so short each day. You have to really capitalize on the periods when they are alert. We did this successfully with 3-month-olds. Going younger than that would require a lot of flexibility and patience, I think.
Thank you.
Okay.
All right, thank you.
Thank you. Thank you. Now, our next speaker is...
You started it?
No, you're okay.
No.
No. I mean, it's her presentation. It's still on.
Yeah, you just press this.
Oh, okay. All right. Thank you.
Okay. The speaker now is Anirban Mukherjee, and he works as a graduate student in Richard Bone's lab in Florida. So over to you.
So, good afternoon, everyone. This presentation follows on from the one given by my advisor, Dr. Bone. Today, I'll describe the repeatability of MapCat SF single fixation, which is a new LED-based flicker photometer for measuring MPOD. It is a novel device that takes into account the aging lens. We wanted to see what would be the effect on MPOD if the age-related change in the lens were not taken into account. To test the repeatability, we recruited 53 untrained subjects and tested and retested on the same day. In the Bland-Altman plot on the left, we plotted the, In the Bland-Altman plot to the left, we plotted the difference between the test and retest MPOD against the average. The average difference between the test and retest was 0.003, and the 95% confidence limits were at plus minus 0.083. A regression analysis of the scatter plot on the right gave an R-squared value of 0.95. Now, from various researchers on the variation of MPOD with age measured using HFP, some have reported an age-related decline, some a rise, and some no change at all. The MapCat incorporates the change in luminous efficiency function with age, based on a model that attributes such changes to the progressive yellowing of the lens. We wanted to test if there is an age-related effect on MPOD if the aging lens is not taken into account. We simulated an experiment by generating random numbers. By generating random numbers to represent subjects of ages 20 to 90 with MPODs between 0.0 and 1.0. These are represented by the black dots, and as you can see, there is no age dependence. These random numbers were used to calculate the flicker null settings for the foveal and perifoveal tests that would be obtained using the MapCat. These were then used to calculate the peak MPOD, assuming a standard luminous efficiency function that is the same for all subjects. These are shown by the red dots. As a result, we found an apparent decline in MPOD with age to be 1.44 plus minus 0.51 times 10 to the power minus 2 per decade, comparable to some of the other using LED-based HFP. In conclusion, care should be taken in interpreting MPOD variation with age if acquired by LED-based HFP. Thank you for your attention and the organizing committee for giving me a chance to present my work.
Yet again, nicely in time. You're doing well so far.
Practiced 5 times with Dr. Bone. Credit goes to him, not me.
So that, thank you,
Need
Richard.
to have a good advisor to do good research, actually.
Yeah, okay. But main problem is that we're running out of options for sausages for breakfast. This is a little bit worrying. Now, questions, please.
I have a question, actually. And
Yes.
the reproducibility data that you presented, what was the age spread of that sample?
It's like, it's between, say, it was 20 to 79, 80. That was what we got.
Okay. Any more hands showing? No. All clear? Okay.
Thank you.
Yes.
I was saved of the blade.
Oh,
Yes.
we have, yes.
Yes, ma'am.
I'm not sure that I understand it well enough, so may not be a good question, but I'm thinking I have another device to measure macular pigment by heterochromatic flicker photometry. Are the calculations that you're doing in your study applicable to other devices?
Ma'am, well, I mean, First of all, the MPOD that we are measuring, we are using a 14-degree stimulus, as Dr. Bones said, right, to do the peripheral measurement, and we have a 1.5 degree doing the what you call the foveal measurement. But I mean, like we believe that yes, they are comparable, but since we are not using the same set of subjects, we can't exactly like say that, or rather give a comparative performance result.
Okay. Yes.
So I just wanted to make the point, and I think this is a good general point just to make at this meeting, and that is that it's likely the case that no two machines, even made by the same manufacturer, are comparable.
Right.
And irrespective of spatial width. And the reason is because the measurement is very attuned to the bandwidth of the LEDs.
Right.
And so when you buy these LEDs, they're always different, a little
right.
bit.
That's what actually I'm doing as a part of my dissertation. That is like, even if you give a nanometer shift to the LEDs, right, we see a prominent difference in the measurements that can be affected, and it's mostly affected by the green LED, not the blue LED to that point. That's what we found.
Okay. Thank you. Thank you. And now we have a speaker from the People's Republic of China. Now, can I get this right? Jinda Zhu. Jinda Zhu. Yes. Thank you.
Hello, everyone. Nice to meet you in Acabei. I'm Jinda Zhu from ZMC China. It's my honor to share with you our study on the toxicity of Mr. Zeaxanthin in rodents. As we all know that Mr. Zeaxanthin is an important centrophile member family of carotenoids, and it is isomers from lutein. And it has related high concentration in macular and believed to play an important role in human eye health. And it is necessary to investigate its toxicity throughout. In this research, we provide a complete assessment of toxicity of Zeaxanthin, Mr. Zeaxanthin, and through acute oral toxicity study and general toxicity study, as well as 19 days subchronic toxicity study. At last, we found in the acute oral toxicity study, both SDRS and other mice has over 10 gram per kilogram dosage. And in the 3 turns of general toxicity study, the results are all negative. And in the 19 subchronic toxicity study, groups with dosage of 1,200 and 600 milligram per kilogram, and the level CS macular degeneration, while 300 milligram per kilogram were tolerable. So I will conclude that for Mr. Zeaxanthin in rodents, it has no acute toxicity and no noticeable general toxicity. The no observed adverse effect level is 300 milligram per kilogram. And after the application of 100 factor safeties, and we obtained the RDI value is 3 milligram per kilogram PW. However, to further confirm this finding of no toxicity of Mr. Zeaxanthin in the application of human consumption, a long-term animal test, where human clinician traveled safety evaluation were suggested. This is my composition. This is presentation. Thank you very much.
Yes, we have hands going up for questions. I think Julie, you first.
I'm wondering what type of a difference you were powered to detect in the just choose one endpoint, one toxicity endpoint, and how powered, this is a typical problem with clinical trials, is not being powered enough to detect a negative, a toxic effect. And so I wonder, in your rodent study, do you have a feeling for how variable the endpoints are and how that influenced your statistical power?
The power is suspended in oil.
question is, with humans, the criticism is that you may not have enough of the sample. Like, what's a meaningful endpoint? So how does that reflect in the mouse model? Is that correct? Yes.
Sorry, I'm not happy with my listening English.
就是说，如果你有不同的那个就是研究不同的结果的时候，你研究不同的结果对你的这个研究的这个 power 就是有什么样的影响？就是如果你看不同的结果对你的 power 有什么样的影响？它这个问题，就是你看你不是有结果吗？不同的。
How many mice? Like how many times did you repeat the experiment? How many mice were used? Maybe that information would help the answer.
How many mice would you need
to
see a toxic effect if one
how many
is
mice?
present?
In different in acute toxicity and general toxicity, there are different amounts of mice.
Okay. Thank you. And we have one more.
We heard this morning that mice have the ability to break down these carotenoids very quickly. So, can these results tell you anything at all about human toxicity?
鼠能把那个 carotenoid 就是分解非常快就分解，所以说你这个结果鼠的结果能不能用在人类身上
？I have said that to further confirm the safety, the toxicity of zeaxanthin in human consumption, and a long-term animal test or human clinical safety assessment evaluation should be suggested. This is results in rodents, just in rodents.
I think long-term evaluation of all carotenoids would be important.
Yeah.
We've looked at it for 3 macular carotenoids for 3 years now, but I do agree long-term is essential as well. I think all carotenoids are something.
Mice are probably not a good model.
Yeah, but I think it's an important contribution to it.
Yeah. Okay. Thank you. Thank you as well. And thanks also to our next speaker for helping with the interpretation during the question session. Our next speaker is Joanne Wu, who is from Boston, United States. And now I'll hand over to you. Remember, this is English now, not Chinese.
I know. I'll stick with English throughout. Okay. Good afternoon, everyone. Today my talk is focusing on dietary omega-3 fatty acid with the risk of AMD. First, I'd like to give you a brief introduction on why we are interested in this nutrient. As you know, DHA is a structural component of the photoreceptor outer second membrane. The tissue level is modifiable by the dietary intake. The biochemical properties, including anti-angiogenesis and anti-inflammation, and all this support the importance of this nutrient in the prevention of AMD. We conduct our study in 2 large US prospective cohorts, the Nurses' Health Study and Health Professional Follow-up Study, with a total sample size of over 100,000 participants and 20 years of follow-up. We used a validated food frequency questionnaire to have repeatedly assessed 2 types of omega-3 fatty acid. The long-chain omega-3, which include DHA and its precursor EPA, and coming from marine source, and also the short-chain omega-3, the alpha-linolenic acid, which I will abbreviate as ALA in my following presentation. It's primarily coming from vegetable source. In our results, we find that both DHA and EPA are significantly associated with lower risk of early AMD. However, we do not see any protective association for late AMD. In contrast, ALA is significantly associated with higher risk of early AMD. Next, we went on to assess the major food sources of DHA and EPA and to see how that relates to risk of AMD. We find that those with high consumption of canned tuna had about 38% lower risk of early AMD. And high consumption of canned tuna is protective for late AMD as well. In contrast, white meat fish, which doesn't include that significant amount of DHA and EPA, is not significantly associated with either type of disease. In conclusions, our perspective data suggests that the long-chain omega-3, DHA and EPA are associated with lower risk of early AMD. And higher consumption of dark meat fish may lower risk of AMD. The association between ALA and early AMD may deserve further investigation. Thanks. I'm happy to take any questions.
John.
Hi, you've got a large enough group that you might stratify versus racial identities. Have you seen any differences in the outcomes in different racial groups?
Sorry, I forgot to mention that 97% of our population are white, so we actually do not have the power to stratify by the racial differences, but that's a very good point.
So I have a question here. How do you think your data, do you think it's consistent with the ARID study, and or would you like to comment on the ARID's finding that they didn't find an effect of omega in?
They did find an association between omega-3 and AMD, but they find a strong association with the dry type of advanced AMD. And now our data is, that's why we didn't find association with in our late AMD, 90% are neurovascular AMD. That's why we did see an association. In A-rates, they see an association between DHA and the central jugular atrophy. And then because they're doing, looking at the progression from early AMD to late AMD, that's why they do not have data on early AMD. So our data is actually on the incidence of early AMD. So overall, I think our results are broadly consistent with the A-rates trial. Okay, thank you very much.
Thank you.
Thanks.
Very good.
Yeah. Moving on, number 6. Our next presenter is Amy Low from Hong Kong. Yes?
Thank you.
Yeah.
Okay. As you all know, retinal ischemia reperfusion injury is associated with many eye diseases, and that will damage the neurons, and eventually it will lead to permanent visual loss. So here we would like to explore the neuroprotective effects of lutein, which has been shown to be quite beneficial in AMD patients. So in our experiments, we occluded the ophthalmic artery to induce ischemia to the eye for 2 hours, and then the occlusion was removed to allow for reperfusion. And lutein was given 1 hour before and 1 hour after reperfusion. And then at the end of the reperfusion, we did the following analyses, including the ELG responses and some structural analyses of the retinal sections. So for the ELG responses, as you know, as you can see, ischemia reperfusion injury greatly affected the waveform. But then with lutein treatment, that waveform is almost completely restored. And then morphologically, IR injury will also induce significant cell loss, as indicated by the presence of the empty space and pycnotic nuclei in the retinal sections, as shown in panel B and panel F here. But then with lutein treatment, as you can see, the retinal morphology was almost preserved. We also further confirmed this study by counting the number of tunnel-positive nuclei. And again, with lutein treatment, lutein can significantly reduce the number of apoptotic nuclei in both the GCL and the INL. So besides saving the neurons, the lutein treatment can also reduce gliosis in the GCLs, as indicated by the downregulation of, or the decrease in the upregulation of the GFAP immunoreactivity. Also shown here is the hypertrophic Muller cell processes that are very prominent in the ischemic retina. But then this phenomenon is also decreased by the lutein treatment. And since Muller cell is a major source of pro-inflammatory cytokines release in the retina, so we further looked into the response of the Muller cells towards ischemia. Here we used a cell line, the red Muller cell line, and exposed the cells to cobalt chloride-induced hypoxia. As you can see, the two pro-inflammatory cytokines that we looked at, that includes the IL-1 beta and COX-2, both of them are upregulated by the ischemia by the hypoxia, but then that increase, that upregulation was decreased by the lutein treatment. And similar results can be obtained when the cells are exposed to hypoxia in a hypoxic chamber. Okay, yeah, okay, I'll skip that. So I hope here I have enough data to show you that lutein is beneficial, and I hope to convince you that lutein can be as a neuroprotectant by its anti-inflammatory, anti-apoptotic, and antioxidative effects. Thank you. Thanks to organizers.
Thank you.
Yep. Questions?
Now, any questions for this paper, please? Yes, John.
So you're seeing decreased transcription of these proteins. So how do you propose that this is happening? What's the mechanism that you think might be involved?
Yes, that's the stuff that we are working on now. Because we are focusing our attention on the NF-kappa B pathway, which I did not really talk about here. But so that we are hoping to see if lutein will be involved in this pathway, maybe having effects on the IKK kappa B, IKK kappa. So that's one thing that we are looking at.
Yes, one more, David.
Can you tell me, did you measure the lutein in the blood of the animals?
No, we tried, but the level of lutein in these animals are so low. We tried to use the HPLC, but then with our own eyes, we can see the orange color in the extracts before injecting that into the HPLC. But it's way below the detection limit of the machine, and we are not getting any signal. Because here, the dosage we use is actually based on the ones that is now used in human. So this is 0.2 milligram per kilogram, so which is really, really low in here. And we cannot detect. But I know that you might have a question that is can the lutein actually get into the injury site? So remember that this is an injury, and there is breakdown in the blood-retinal barrier. And lutein is very, very lipophilic. So we anticipate that the lutein can actually get into the injury site because of the breakdown in the barrier. And we've done similar things in the brain, and then we can see that in the brain, there's also breakdown of the blood-brain barrier, and we anticipate that lutein is also doing similar things in there, near the injury sites.
Thank you.
Thank you.
Now we're staying in the East with this very international collection of speakers that we have this afternoon. And our next presenter, Akira Obana, is from Japan.
Yes.
Okay,
Thank you very much. I guess some people in this room are a little bit skeptical to the resonance Raman spectroscopy, especially concerning the age effect, because there is a possibility that a large contribution to the there is a possibility that a large contribution to the decline with age by resonance Raman spectroscopy is due to an artifact from cataract. Then we investigated the age effect on NPOD levels with RRS in the eyes with clear intraocular lens implantation in order to avoid the effect of cataract. Subjects 144 eyes, all are Asian, and no fundus diseases, and no history of routine supplement taking. These are the results. The NPOD ranged from about 800 to 12,000, and the median was about 4,000 Raman counts. And these tables are the most important results, and we found two factors that were significantly associated with NPOD levels. This means age is one factor. This means if you're getting old, 1 year old, then the NPOD declines 59 Raman counts. And also the actual length is the significant factor. So this means MPOD levels decline more than 10% each decade. So in conclusion, age was a factor that associated significantly with low MPOD levels. MPOD levels decline more than 10% getting 10 years old. Axial length was also a negative predictor of MPOD. Other factors exhibited no significant relationship with MPOD. But there is a limitation in this study. The present study included patients only aged 50 years old, 50 years and older. And the amount of intake of lutein and zeaxanthin was not included in explanatory variables. These are the limitations. Thank you.
Thank you. We have a question here.
You showed that pupil diameter influences your measurements a lot. Did you correct for this?
Yeah, we measured the pupil diameter, and all patients' eye has more than 7 millimeters. 7 millimeters and more.
Also the elderly, because pupil diameter for the very old, that's a lot.
Yeah, but we use the topical eye drops. And this is, most patients are 1 day after the operation, and they have almost this very good mid-races. Otherwise, we don't operate so much.
Any more hands? Yes.
Thanks. It looked like you had a very strong effect with axial length.
Pardon?
And with axial length.
Axial length? Yes, it is a significant factor.
Right. So that longer axial lengths were associated with lower macular pigment optical density. And
if the axial length becomes 1 millimeter, then about 400 declines in about 400 Raman counts decline.
It's a huge effect. Yeah. Okay. Yes. I'm just wondering if that could be due just to sort of stretching of the retina, but it sounds like it's a much bigger effect than you could account for that way.
Pardon?
You know, stretch the retina, then you're sort of diluting the macular pigment, but it sounds like a much bigger effect than that.
I don't know the exact result.
Okay. Thank you. Thank you. Now we change to a different kind of topic and a different geographical location, and Tim O'Hare has come all the way from Australia for this for us. Thank you, Tim.
I actually won the travel grant to be here.
Yeah.
Thanks very much. Thanks for the travel grant too. That was very helpful getting here. But I'd just like to talk very quickly about a 4 or 5 year project that we've had on increasing zeaxanthin in sweet corn. We all know that lutein is a lot more common in the diet, but zeaxanthin is a bit rarer. So the aim behind this project was to actually create, through conventional breeding, a sweet corn cobet that would give you 2 milligrams of zeaxanthin as part of a normal meal. And down in the bottom corner here, there, that's the 2 milligrams per gram per person per day sweet corn. The reason we chose sweet corn was that it's already one of the best sources of zeaxanthin. But the problem is you'd still, even with this high level, you'd have to eat somewhere between 4 and 10 cobs per day to get the 2 milligrams. So we thought it was a good target for biofortification because it's a tasty thing. You can put it on your plate, and to increase bioavailability, you can put a nice big dob of butter on it and not feel guilty about it. Okay, so yellow sweet corn, when you look at it, the major carotenoid is lutein over there. So the ones in green are the carotenoids that you tend to find in sweet corn. Lutein makes it yellow. This side tends to be more orange. So these carotenoids, when you increase them, you start to change the colour of the corn itself. So we had 2 breeding populations, and with one of them we increased Firstly, the beta arm of the pathway. So we shifted, instead of the carotenoid flux going this way, we shifted it this way, and by about 3 or 4 times. And in the second population, we increased total carotenoid production by about 2 to 3 times. So all the carotenoids went up. One way of doing this, we did it with HPLC, but we did a screening method to begin with. Changing carotenoids changes colour. And so over here we have hue angle. This is an objective measure of colour. Anything with a hue angle above 83 we got rid of. And we analysed these by HPLC, brought them over here. And you can see the switching around of the different peaks. You probably can't see that from where you are, but lutein and zeaxanthin, zeaxanthin is much higher down here than the lutein, and we could quantify everything. Just a quick point over here, when the correlation, not too bad, 84% regression, if you include zeaxanthin along with the minor carotenoids, beta-cryptoxanthin and beta-carotene. In conclusion, what we did then was we actually added 2 together. The high carotenoid ratio with this one over here, the high total, we slapped them together and we doubled the carotenoid level again for zeaxanthin. So we ended up with this, turning this into this and changing the colour of the corn, which is actually, is a good thing for market differentiation, so that people can actually go out and buy their sweet corn and they know that they're getting a high zeaxanthin line. So now we have a half a cob of corn that will give you what arids to give you daily as a supplement. So you'll get it as part of a normal meal. So.
Yeah. Yes, we have one here.
Have you looked into what the cost would be to produce that on a mass scale and be able to actually have production of that enhanced corn with zeaxanthin?
Well, the answer to that is that it would be like normal sweet corn. The cost of it, the way it would be marketed would be through a closed loop system, so that it would be priced at a premium. It wouldn't just flood the market with high zeaxanthin sweet corn. So there has to be some benefit to the people who we have to control the level of zeaxanthin in the corn and make sure that consumers are getting what they're paying for. So.
Is the zeaxanthin in your corn more readily available than pure zeaxanthin? Because you know in the egg, of course, it's about 4 or 5 times more available.
Yes.
Have you done blood studies and see if it actually increases the blood greater than you would expect?
No, but this is the next step. We've just done about 150 experimental hybrids. Of course, you need to put together 2 parents to have the hybrid vigor in the corn. We expect it would be, as long as you macerate it when you eat it, it doesn't go straight through you, and perhaps have some lipid with it, we expect it would be a pretty good matrix as compared to things like lycopene and crystalline carotenoids. So no, that's the next step. So we say that in our abstract, that we haven't compared supplemental zeaxanthin with zeaxanthin from the corn. And in fact, the level of this sweet corn is actually a bit higher than 2 milligrams. It's about 2.5. So I suppose, it doesn't hurt. We don't want to make our corn too orange, though. I mean, that can turn consumers off. So it's a product we eventually the consumers want to buy. They don't want to buy something that's too orange.
Yeah, we want to encourage participation by the other speakers, so we can take one more quick question.
Mine's quick, and this may be a dumb question. I know that sweet corn and the corn that makes popcorn are 2 different plants, but could this be translated into something for, I'm thinking of kids eating popcorn. Could that even be a possibility of making zeaxanthin available through a food like that?
Yeah, you can do it. I mean, there's a lot of flint maize lines that you can do it. You could take it back into popcorn. As long as the genetic diversity is there. A lot of the diversity here actually comes from Caribbean flints, which is different. So sweet corn, I mean, I could talk about it for ages, but it comes from Illinois, the actual shrunken 2 sweet gene, so that Jim Brubaker in Hawaii had to bring in the Caribbean flint to adapt it for tropical climates, and that's where the colour comes from. So.
Okay, thank you.
Okay, thanks.
Laura Fletcher. Yeah, okay. So we're skipping one, yes. Yes, okay. Yeah. Now we're not doing poster number 9, we're moving on to number 10, which is going to be presented by Laura Fletcher, another member of the Hammond Lab.
Okay, so I'm going to talk about some of the more optical properties of macular pigment, specifically the visibility hypothesis. And I wanted to use this picture specifically because often when we're viewing objects in the distance outdoors, we're looking through this shortwave heavy haze. And we've talked before already today about how macular pigment absorbs shortwave light. So the idea here is that the more macular pigment you have, possibly you could extend your visual range outside, which is essentially what this says. And there's been some prior modeling that's shown for individuals with equal Snellen acuity, those with high macular pigment could have up to a 30% farther visual range than someone with little to no macular pigment. So I measured macular pigment using HFP, and we used just the standard 1-degree stimulus size. And then we did ascending and descending method of limits assessments for a 7.5 cycle per degree sine wave grading target. And we did this in the presence of simulated haze and also with just broadband xenon light, in addition to a shortwave deficient background so it would not be absorbed by macular pigment, to basically spectrally obviate the effects of macular pigment, thus showing that the other two conditions are likely driven by macular pigment. So the results here, you can see this graph on the left is the haze log energy at thresholds. So this would be the average of all of the transition points for the ascending and descending thresholds. And we have a pretty strong correlation between macular pigment and the energy in the haze background. We see the same relationship for the broadband xenon background. Xenon light has a lot of shortwave, or xenon, the spectrum has a lot of shortwave energy, so we expected to see that same relationship there. Macular pigment was not significantly related to the energy in the background for the thresholds at the 558 nanometer condition, which is what we expected. The average macular pigment for our sample was pretty high. It was 0.46 at 30 minutes retinal eccentricity, but the range was pretty large. It was ranged from 0.15 all the way up to 0.91, and we had 25 subjects. So basically, haze log energy at threshold varied by a factor of 2 between the people with the very lowest macular pigment and the highest macular pigment. So this sort of suggests that if we're taking this as a proxy for more of this energy in the background being a greater distance between the observer and the target, this would translate into a pretty significant difference. We saw the same effect for the xenon, and again, no effect for the 558 as expected.
Thank you. Now we're open for questions again, please. Yes, Julie?
I was wondering, how much difference do you expect that to make in real life situations? And which people or which demographic benefit most from that kind of work?
I would think anyone who's pretty much performing a job outside, so maybe pilots or snipers, anybody who's hunting, so anytime you're doing something at a long distance in which you need accuracy.
Do you think there's like a magic number of macular pigment that you'd like to have? If you know, patients will always ask the clinicians, well, what level do I need to get to? Based on your experiment, do you think it's different for everybody, or do you think there is a magic number of macular pigment?
I'm not sure. We've done one previous study from our lab showed that there was a plateau effect after about 0.6, and I know there's been a few other studies showing some of these different effects of macular pigment plateauing, but I did not see that in mine.
Yeah.
I'd like to sort of continue to extend this, maybe get more people and see if with a greater sample size I would see that.
Yeah, I mean, I think the interesting point behind that is that the average macular pigment is much less than that.
Yes.
For that central eccentricity.
Mm-hmm.
So it means that based on what you're saying, that, okay.
Yeah.
Thank you.
No snipers in the audience with a particular interest who'd like to ask something?
I'm not advocating, just saying.
Okay. Okay, thanks very much. Thank you. Now we're staying in the same lab with a different speaker. This time it's Emily Bovia.
So today I'm going to talk to you about critical flicker fusion thresholds and the relation to neurocognitive function. And this project is based on the idea that lutein and zeaxanthin can promote neural efficiency. And supporting behavioral evidence thus far comes from relations between macular pigment as a marker of cortical lutein and zeaxanthin and relations with temporal vision and cognition. Now, these relations likely reflect a similar underlying phenomena, mainly speed of neural processing. So the purpose of this project, firstly, was to determine the relationship between CFF thresholds and neurocognitive assessments. And in the second part, we wanted to determine the effects of lutein and zeaxanthin supplementation on CFF and cognition. So to look at these two things, we pulled data from 45 subjects, young adults aged 22 years, who were enrolled in a one-year supplementation trial. For the first part of the baseline comparisons for the CFF and neurocognition, we selected 15 subjects with the highest CFF and 15 subjects with the lowest CFF. And for part two, as a preliminary look at the effects of supplementation, we selected subjects who had at least a 1/10 of a log unit change in macular pigment as assessed by HFP. CFF was assessed with a customized LED device that was configured to obviate the influence of macular pigment. And neurocognitive function was assessed with the CNS Vital Signs Brief Core Assessment. And this looked at specific cognitive domains such as executive function, psychomotor speed, memory, and so on. So results for part one, subjects with higher CFF performed significantly better on tasks related to motor skills and rapid decision making. So the three domains that we see significant effects at are executive function, psychomotor speed, and cognitive flexibility. So subjects with high CFF are performing at about the 75th percentile, where subjects with low CFF are at about the 55th percentile. And for part two, significant increases in CFF and these three cognitive domains were found for those subjects who had an increase in macular pigment of at least a 1/10 of a log unit. So just to summarize, in part one, we did have this significant association between CFF and neurocognitive function, which is suggesting that these past associations with macular pigment are likely reflecting that speed of neural processing. And also we can consider the utility of CFF as a measure of processing speed. And in terms of part two, our preliminary data indicate significant changes in CFF and neurocognitive function after supplementation with lutein and zeaxanthin. So in general, our results lend support to this neural efficiency hypothesis of lutein and zeaxanthin function, and also support to behavioral methodology to assess neural processing speed and also test the effects of supplementation with lutein and zeaxanthin. Thank you.
Anyone with questions, please? Yes, right at the back there, please.
Does your research respect Bloch's law, or does it need to be rewritten?
I'm sorry, I'm having a hard time hearing you.
Sorry.
Does your research respect Bloch's law, or does it need to be Bloch's law need to be rewritten?
Bloch's law with respect to the assessment of CFF?
Yes.
Yeah, so that is configured to account for that as well. Mm-hmm. Actually, this is a part of a larger scale project. So we have data right now from our younger subjects, but we're also looking at patients who are healthy elder adults and patients with MCI. So that's still in the preliminary baseline stage, so we'll have CFF data on that as well.
Okay.
Thank you.
Thank you. Now a change of topic again. We're going back to food. And as you can tell from Antonio's profile, he's a great expert on food. Antonio Melendez from Sevilla in Spain.
All right. Thank you very much. Good afternoon. In our study, we just categorized fruits and vegetables according to their content on lutein and zeaxanthin. This study is was just made in the context of International Network on Carotenoids and coordinating. We are so far over 45 groups from 15 Ibero-American countries. We received funds from CITED, and one of our main objectives is to find new sources of beneficial carotenoids among native products. So our study can be considered like a follow-up of a previous work in which we screened the content of lutein and zeaxanthin in up to 74 Panamanian fruits and vegetables. What have we done? We have selected over 30 references. We have looked at the lutein and zeaxanthin levels, and we have categorized them following the criteria proposed by George and Fred Kachik, which I think it's really useful. According to them, for a source to be considered a very good source of a carotenoid, it should contain above 2 milligrams per 100 grams fresh weight. So what were some of the main problems we found in this kind of review? One of them, as we all know, the content in carotenoids is dependent on so many factors, like genotypal, agronomical, climatic, etc. So that's why we believed more in the minima and maxima values than on the average values. We took those into account, I mean, minima and maxima. As we all know, in some older studies, there wasn't a distinction between lutein and zeaxanthin, so that was a problem. And another problem is that in some cases, the reported data are little reliable. So this is a summary of our study. In the case of lutein, we looked over 130 vegetables. And as it's well known, we found so many very good sources. As John here and George said before, leafy vegetables are very good sources of lutein. In the case of fruits, the situation was different. We found only very few that were good or very good sources of lutein. What happened in the case of zeaxanthin? We didn't find any very good source in between vegetables, but we did find some new sources and very good sources of zeaxanthin. This is important. I mean, up to some years ago, there were only 3 very good sources, like sweet corn, orange pepper, and Chinese wolfberry. So in our study, in Panamanian fruits, we found 4 more. All right? This is really important because these are not well-known fruits. They are not even domesticated in some cases, although they've been consumed by the Aborigines for centuries now. Among them, I want to highlight Sastra because it's at the same time a very good source of zeaxanthin and also a very good source of lutein. So I think it's important for macular researchers and companies. Thank you very much.
Thank you, Antonio. I would just like to comment on one thing, and that is the network, the carotenoid network that Antonio is coordinating for carotenoid research groups in Spain, Portugal, and the whole of Latin America. This has brought so many groups together, and it's a good example of what can be achieved if somebody really does want to construct such a thing. And it's going to take our carotenoid research, especially in the food area, forward a great deal in future years. So anyone from other parts of the world, think about this as a possibility. Any questions, please?
What are the levels of zeaxanthin in these 4 fruits that you mentioned?
They were really, really high. In some cases, over 40 milligrams per kilogram. I mean, over 4. I can give you the data later, the exact figures. But very, very high.
John, one here, please.
There are two angles on this. I mean, there's the industrial angle of extracting the carotenoids, and then there's the desirability just to eat the fruits. So how do they taste?
I haven't tasted them, but I've got reliable reports that they are tasty. And I think, I mean, they are tolerable. And actually, they are good. They are good. All right, but it's really interesting. I mean, there are so many unexplored fruits in South America, and they are really good sources of not only carotenoids, but also another health-promoting secondary metabolites like acai and things like that. So I think it's a research field that deserves encouragement. Yeah. So they are edible. I can give you some if you want.
Okay.
All right. Thank you very much.
Antonio.
Thank you.
Now we're staying in South America. Our next speaker from Brazil is Simone Acabei de Oliveira. Yeah. So over to you.
So, good afternoon, everybody. Yeah, I'm going to talk about the development of extraction process of xanthophylls lutein and zeaxanthin from Brazilian maize landraces. So, maize is the third most cultivated cereal in the world. In Brazil, is the fourth largest. In Brazil, is the fourth largest producer. So.
Here you go. You can hold that up.
Thank you.
No, it's okay. It's a sore throat, so just give her a second. Now go with that. You're going to have to hold this, though, okay? Okay?
Thank you. It's better. So, maize landraces are local varieties that has high adaptability to the environment, so that they need no agrochemicals use or just a little. Our research group has detected and quantified carotenoids in maize seeds and leaves. But the extraction method was based on the conventional one that makes use of organic solvents that usually leaves residues after evaporation that are toxic for humans and for the environment. So, age macular degeneration is the major cause of blindness in the world, and in Brazil, around 6% of the population suffer with this disease. The xanthophylls lutein and zeaxanthin are in high concentration in the retina, and looks that they prevent the AMD development due to their antioxidant and antiogenic activity. And that's why lutein capsules are widely used for AMD prevention. They are made of these species of flowers, and in Brazil, the raw material of these species and the lutein capsules are fully imported. So the main objective of my work is to answer these questions. Which local landrace of maize has the highest concentration of carotenoids in which part? If and which edible oil can better extract these carotenoids, and which energy source. So for this, leaves and seeds, flowers from leaves and seeds from 5 maize local landraces were incubated with 10 milliliters of edible oils in the dark and irradiated for 30 minutes with ultrasound and microwaves. And the carotenoid content was measured by 450 nanometers, and lutein identification was by HPLC. And the edible oils used in this work was these ones. And these pictures show the carotenoids content in the distinct 5 varieties of maize, in leaves, irradiated with microwaves and ultrasound. And this picture shows the seeds irradiated with microwaves and ultrasound. And putting this together, we saw that the leaves of the landrace called R8C, incubated with canola oil, was and irradiated with ultrasound, was the best carotenoid structure. And this oil resin, the main carotenoid compound was lutein, as shown by HPLC. We did a patent request to the National Institute for this method request. The leaves are waste, so this is interesting to add value to a disposable raw material. And next step is to scale up this method for capsules production. Thank you, everybody.
Anyone with questions, please? Yes, Neil. The mic's coming round.
Were you able to determine if you had esters of the xanthophylls in your extraction process? And was that carrying over into the canola oil? Because your HPLC showed free lutein.
Yes.
Had you esters?
Did you find
Were you able
esters?
to determine esters? Was there esters present?
Esterified.
I don't know. I haven't measured. Actually, in leaves, I have to saponify, but in seeds, we don't have to saponify for HPLC analysis.
It's very rare to find them esterified in leaves, so I would expect it to be free in leaves.
Okay.
We have one more, Katie.
Sorry?
How easy is it? Could this method be for scaling up? Yeah.
Yeah, I think there is no problem because there are microwaves and ultrasounds with high capacity. We don't have in our lab, but we know an industry in my hometown that they have large capacity microwaves and ultrasound. Yeah, but before to scale up, we have to do kinetics studies that we are planning to do before to go to this step. Because yeah, if it's this oil resin made of enriched with lutein is not like the conventional one, we don't have a work.
Okay. Thank you.
Thank you.
Now, our next speaker is Raymond Byrne from University of Ulster in Northern Ireland. Raymond.
Good afternoon, everybody. My title of my poster is The Macular Pigment, Optical Density, Spatial Profile and Increasing Age. And we've already heard a little bit today from various studies on how macular pigment might change in the normal aging eye. And that there's different studies have presented different results, which might depend on the instrument used, the population studied, dietary intake, and a variety of other factors. And I currently use a HFP-based device, and one of the limitations on the age studies in the literature at the moment is that most of the studies report information from just that one location that was mentioned this morning, that magical 1 degree diameter stimulus. So what I wanted to do was to do an age-related study, trying to measure the spatial profile in normal eyes to see if there's a little bit more information out there that we need to know about. So I had 98 Caucasian individuals aged between 19 and 71, and they're all screened for ocular normality. assessed as healthy and as normal as one can possibly do. I measured macular pigment at 4 different retinal eccentricities using Billy Wooten's instrument. What I found was that for 3 of the 4 retinal locations that I measured macular pigment at, there was no significant association with the age. But at the furthest eccentricity at 1.75 degrees, there was a small but statistically significant positive association between age and macular pigment, which, interestingly, when I broke it down into gender, seemed to be due to just the male population. So overall, found that there was no significant association between macular pigment and the age for 3 of the 4 locations measured, with a clinically insignificant increase at the furthest eccentricity. And while I was carrying out this age study, several studies in the past reported variations in the spatial profile within the central macula of the macular pigment itself, so that participants don't necessarily have an exponential decline in the macular pigment from the foveal center. And what I found in my study was about 15% of the participants showed a non-exponential profile. And I think that's important, particularly for supplement studies, when we're reporting or trying to look for changes in macular pigment over time with supplement, or even with the age, that we try and present information in more than one spatial location if possible. Thank you.
I think that's the record, 2:59. Questions, please? Yes, we have one over here.
Hi, I was wondering...
Can you get the microphone, please?
Hi, I was wondering if these atypical spatial profiles, if you found any relationship with age as well? Or can you go a little bit more into detail on that?
In my study, no, I didn't. There was no age difference between the typical and atypical profiles. I think John had found in the past that it was more older individuals, but I actually had a relatively small sample of, atypical profiles. So.
Okay, and gender?
No, there was no other pattern, but again, as I said, it was a small number, so.
We heard this morning that, in fact, in a very large study, there was an effect of age. Do you think the reason you haven't shown effect is because your numbers are small? I mean, what's the reason?
It's potentially a reason. I think my study is limited by numbers, but also, to have serum levels or dietary intake. The majority of studies for HFP-based methods show no significant change with age, although there's a significant amount of studies that show a small decline with age as well. But slightly more studies overall would show no change.
Okay. Thank you.
Thanks.
Thank you. Yep. And now we're down to the last speaker before the break. And this speaker is O.A. Mahru from King's College London.
Thanks. So many of you might be familiar with the first of Tolkien's trilogy, The Fellowship of the Ring. I'm here to talk about the book that Tolkien didn't write, The Heritability of the Ring, or more specifically, heritability of the ring-like distribution of macular pigment assessed in a twin study. And I should say at the outset that a lot of the analysis was done by a master's student, Ambreen Tariq. It's been suggested that a ring-like distribution of macular pigment may be associated with a reduced risk of age-related macular degeneration, and much of the current research we're hearing about focuses on modulating environmental factors such as diet and seeing whether that might have an effect in preventing AMD development or progression. So it's of interest to know how much of the variation in particular pigment profiles are related to genetic factors and environmental factors. And one amenable way of doing that is using a classic twin study. So that's what we did. We tried to quantify how much of the pigment profile or the ring-like pattern, the presence of the ring-like pattern, could be explained by genetic as opposed to environmental factors. So in 314 healthy Caucasian twins, macular pigment optical density was measured using 2-wavelength autofluorescence. These twins were part of the Twins UK cohort, which is based at St Thomas's Hospital. 88 pairs were monozygotic and 69 dizygotic. We took the right eye profile from each twin, and this was assessed independently by 2 graders using common criteria to see whether a ring-like pattern was present or not, and concordance was calculated as shown for monozygotic and dizygotic twins. So this is what we mean by the ring-like pattern. On the left, you have the 2-dimensional optical density map showing a central zone of high fluorescence and then a surrounding annulus also, and that translates to, when you plot density against eccentricity, a second peak at about 0.5 to 1 degrees eccentricity. So the overall prevalence of the ring pattern in our population was found to be 21 or 26% for the 2 graders. Interestingly, the case-wise concordance was for monozygotic twins was over twice that found for dizygotic twins, suggesting that genetic factors are extremely important. So in conclusion, the fact that the concordance in monozygotic twins was over 2 times the dizygotic concordance suggests that genetic factors are important, and a preliminary heritability calculation suggests that additive genetic factors could contribute to about 84% of the variance in the prevalence of the ring-like structure. And future studies can look at what those genetic associations might be and what they might mean in terms of susceptibility to AMD. Thanks very much.
Any questions, please?
Yes, just get your mic light.
Can we get the microphone?
People can't hear us.
Any thoughts on why ring structures happen?
Why they happen? No, I don't know of any. They're present. The prevalences seem to vary between, in some studies, 19% to up to 86% in some African populations. Some people have postulated early diet, and I think also that there's not a unified convention on how you grade the structures as ring-like or not ring-like, which might explain some of the different prevalences. But I don't know of any evidence suggesting.
What about just distribution of the binding proteins?
Right, in terms of why the pigment would be associated like that. Yes, I don't know if there's any evidence that the distribution of the binding proteins changed, but that might.
And what instrument did you use?
So this was the modified scanning laser ophthalmoscope, the Heidelberg Engineering, the one that with the module for pigment analysis.
Do we have one more? Yes, Dr. Howard?
Is your ring-like structure equivalent to what the Waterford group would call atypical macular distribution?
That's interesting. So we haven't done a direct comparison using sort of other psychophysical methods of quantifying pigment profile, but I suspect it might be. So it's not the conventional sort of monotonic exponential decline. It's something different. So it probably would be classified as atypical, but you'd need a direct comparison on the same subjects to see if you classify them the same way in both cases.
Okay. Thank you very much.
Thanks very much.